cell suspension, and you take these barcodes. What these barcodes are you can just think of them as capsules which have sequence of nucleotides tagged at the end, so you can differentiate between one barcode to another. And the way this works is this is a microfluidic chamber. Do people know what microfluidics is? Yeah? Yeah. So the way it works is that you put the cell enzyme here, these barcoded cells here, and you hope that based on the size of the cells and the size of the barcode, each barcode gets engulfed by a cell, or each cell gets engulfed by a barcode. So what you've essentially done is that you've taken this, split it into hopefully single cells, and tagged each and every single cell with a barcode. What that helps us doing is distinguishing one cell from another. Right? And then you take these single cells, these barcoded single cells, and then you reverse transcribe all of the RNA that is present in that particular cell. Reverse transcription is nothing but whatever sequence you had, you make the opposite sequence of that nucleotide. And after reverse transcription, you remove all of the debris from the cell, and then you've prepared what is called a cell library. This is all kind of complicated, but essentially the idea is you take that and you convert it into a suspension where each single cell and all the mRNA that was in that single cell has now been tagged and given an identity. And now what you have is, for each barcode, you have this sequence of reverse transcribed DNA. Right? And once you have this DNA, we clonally amplify that DNA, and then we use this technique called sequencing by synthesis to essentially remake or re-identify the sequence of RNA that was generated from our, that was actually captured, essentially. So take the fragments of DNA present in each and every small capsule. Each capsule is a single cell. Take the DNA, reverse, clonally amplify the DNA, and then remake the actual sequence of the DNA that we had actually sequenced. So all this to say that now what we have